Hey all, this side Vinita Tiwari. Welcome to my YouTube channel Learn Life Science. So in this video, I will be talking about superoxide dismutase activity determination by following the NBT method. So firstly, we will see what kind of reaction generally this enzyme carries out. Then we will step into the estimation method, in which I will be talking about the working principle. Then I will talk about the protocol, means how you can carry out the estimation method, and then lastly we will look upon into the calculation. Now, firstly, if we are talking about the superoxide dismutase activity in plants, means at a point we are talking about the physiological response which is given by plant under different condition. For example, even under optimal growth condition, in normal growth condition, the plant produces reactive oxygen species, but at a normal level. But if you are exposing the plant to different biotic stress, for example, if you are exposing the plants by infecting it with different pathogens, or if you are providing extreme condition, means if you are providing abiotic stress conditions like drought conditions, salinity, or temperature condition, then in such condition the plant is going to show response. It is going to give response by generating reactive oxygen species. These reactive oxygen species are generally the species which are derived from reduction of molecular oxygen. These reactive oxygen species are generally derived from the reduction of molecular oxygen. For example, superoxide radicals, then it can be singlet oxygen, then it can be hydrogen peroxide, or it can be hydroxyl radicals. These reactive oxygen species can exceed its level under these stress conditions. Now, if the plant is generating this kind of response, means these reactive oxygen species are involved in oxidative damage, means they are generally damaging the plant cells leading to their death. On the other hand, they are also involved in cellular signaling. This cellular signaling is nothing but the trigger for defense response. Trigger for defense response. For example, different antioxidant enzymes are present in the plant cells which carries out the reactions to neutralize the conditions, means to neutralize the reactive oxygen species that are produced beyond its level. For example, superoxide dismutase in short form it is termed by SOT. Superoxide dismutase generally carries out dismutation reaction. As you can see in its name by itself, the dismutation reaction is carried out by the enzymes, and the dismutation reaction is carried out on the superoxide anions. Superoxide anions means the charged anions and radicals. Okay, for example, the superoxide anion in here is the reduced form of oxygen, that is the superoxide radical. Now, the other name for superoxide dismutase is superoxide oxidoreductase, means if I write it here, superoxide oxidoreductase. Talking in terms of enzymes, you can see that the oxidoreductase belongs to the first class of enzymes. So you can see that it can carry out both oxidation reduction reaction on on particular substrate. What basically happens that the oxygen molecules that are previously reduced generates superoxide radical. So this is a superoxide radical, and now it is going to be targeted by the superoxide dismutase. Now if we talk about the reaction, then the superoxide radicals in the presence of protons, if you see here, it is going to get reduced to oxygen molecule and a single hydrogen peroxide molecule will be generated. Now what is happening here, basically the superoxide dismutase is neutralizing the radicals which are generated by plant in response. Okay, further this hydrogen peroxide is targeted upon by different enzymes, like for example if we talk about catalase. Catalase is an enzyme which targets this hydrogen peroxide and convert it into or split it into water and oxygen molecule. Okay, now we will talk about the different types of so now we will talk about different types of superoxide dismutase which are generally present in plants. So superoxide dismutase is generally reported to be present in 10 raised to minus 5 molar concentration, and it is present in all the oxygen metabolizing cells. For example, it is present in chloroplast, mitochondria, nucleus, then peroxisome, cytoplasm, as well as apoplast. If you see under normal condition, the superoxide radicals are present in the concentration of 240 micromolar, means they are produced 240 micromolar per second, but under stress condition its level is going to exceed to around 720 micromolar production per second. Okay, it is a reported data. So now talking about its type, you can see the first type is copper zinc superoxide dismutase. The classification is based upon the type of metal which is associated with the enzyme in order to carry out its activity at its optimum level. Okay, so the first form, that is copper zinc form, is the most abundant. Okay, so it is the most abundant class. It is the most abundant class of plant superoxide dismutase. Now it can be present in a homodimeric form or it can be present in a homotetrameric form. It depends upon the localization that where it is present. For example, the homodimer form is generally present in cytosol as well as periplasm. The homotetramer form is basically present in chloroplast as well as extracellular space. Okay, now mainly these copper zinc superoxide dismutase are present in cytosol, chloroplast, peroxisome, apoplast as well as nucleus. The important thing about this superoxide dismutase is that this copper which is associated with superoxide dismutase is responsible for carrying out its activity to its optimum level. Okay, now if copper is absent, means if it is lacking condition, if there is a lacking condition where copper is absent, then it can lead to the enzyme inactivation. Many a times we see that the enzyme requires several cofactors for carrying out its reaction, and if these cofactors are absent, then these enzymes are going to get inactivated. So in this very similar way, copper ions are generally acting as a cofactor in order to keep the enzyme in its active state. On the other hand, zinc is basically involved in providing the structural stability to superoxide dismutase. Okay, now moving on to the other type, that is iron superoxide dismutase. These are generally present in chloroplast, and also it is reported that hydrogen peroxide can inactivate these superoxide dismutase enzyme. Okay, these are also present in homodimer form as well as homotetramer form. Then lastly, manganese superoxide dismutase are also present in plants. These are also present in their homodimer form, or it can be in homotetramer form. Okay, these are generally present in mitochondria as well as peroxisome. Okay, so these were the three types of superoxide dismutase that are generally present in plants. Now, moving further, we are going to see that how you can estimate or how you can determine the concentration or activity of this superoxide dismutase in plants. Okay, so we are going to study a method, that is NBT method, which is given by Charles Beecham and FRIDOVICH in 1971. So if you are studying or if you are trying to calculate the superoxide dismutase activity in plants, means you are providing the plant with different condition. Means, for example, you are studying the response given by the plant under stress condition, which I have previously mentioned. In this case, you can see the difference by comparing the plant from control, that is a normal growing plant, with its treated. It means in which you are exposing it to different stress condition. Okay, you can compare the results from both the plants and you can see the difference in the activity of this enzyme. So firstly, this method, means the method which was given by this Charles and Arvin, they generally created a reaction mixture. For example, if we see that, firstly I have mentioned that superoxide dismutase is an enzyme whose substrate is superoxide radicals, right? So firstly, if you want to determine activity of any enzyme, you are going to need its substrate, right? So that's why the first need of this method is the generation of this superoxide radicals, right? You will have to firstly generate superoxide radicals, then you can expose it with your enzyme in order to carry out the estimation method, okay, by which you can determine the activity. Now, this reaction mixture which was formulated by them consisted of riboflavin, then it was consisting of methionine, then it was consisting of NBT, that is nitro blue tetrazolium. Okay, then if superoxide dismutase is present in your test solution, then it means, for example, in this mixture we are going to consider that superoxide dismutase is present. Okay, present, as well as we are also going to see what happens when there is absence of this enzyme. Okay, so this is initially a yellow colored solution consisting of these three major compounds. Okay, so the reaction generally begins when this solution, when the test tube is exposed to light. When you are exposing this solution to light condition, then the riboflavin molecules, means the first step that occurs, that the riboflavin molecules which are present, these riboflavins generally get excited. to a high energy level, because these riboflavins are having the capacity to absorb light. Okay, these vitamins are having the ability to absorb light, and that's why they get to their excited state. Now, in order to return to their ground state, means in order to get to their ground state, the energy is transferred to the oxygen molecules present. Okay, the oxygen molecules which are present in here now will receive this high energy. Okay, and now what will happen, that the transfer of energy to oxygen will promote one of the oxygen molecule to excited state, resulting in the formation of singlet oxygen. Okay, I hope you are understanding that riboflavin, in order to return to its ground state, transfers its energy to the oxygen molecule, and this results in excitation of a single oxygen molecule, which ultimately results in the formation of superoxide radical. Okay, now what happens, that this singlet oxygen generated will be acted upon by methionine. What will happen, that the methionine will oxidize. The methionine will simply oxidize the singlet oxygen, and itself it will going to convert into methionine sulfoxide. Okay, so in here you can see that the superoxide radicals are now generated. So we have seen the role of riboflavin as well as methionine in the reaction mixture. Now what will happen, there will be two conditions. First, if superoxide dismutase is present in the reaction mixture, what will happen, the superoxide radicals that are generated, they are simply going to reduce. They are simply going to carry out the reduction of NBT to formazan. It is basically the reduced form of NBT, means basically NBT is going to accept electron from this superoxide radical, and it is going to be get reduced to formazan. Initially the solution which was yellow will now be converted into blue precipitate. Okay, if it is clear to you, that you can see that now simply the aim of this overall reaction was the generation of the substrate, means once the substrate is synthesized, it is going to be provided with another substrate to which it can reduce for the analysis of its concentration. For example, in lacking condition, means if superoxide dismutase is lacking, then you can see that the formazan concentration, the formazan which is produced in blue precipitate form, can be spectrophotometrically determined by taking its absorbance, right? You can measure its absorbance. Now this absorbance will be proportional to formazan, as well as the formazan concentration is proportional to the superoxide radicals which is generated, right? Now if we are considering second condition, for example, condition in which superoxide dismutase is present in this reaction mixture. For example, you are providing it with your plant extract. This plant extract is your test solution, in which you are targeting to determine the superoxide activity present in this plant extract. Okay, so if superoxide dismutase is present in the solution, it is going to target the superoxide radicals, and it is simply going to convert it into oxygen molecule as well as hydrogen peroxide by the same reaction. Okay, so now what will happen, that the radicals generated are simply going to get reacted or simply going to get reduced by the enzymes, and this will result in low formazan production. If superoxide dismutase is present in minimal amount, then there will be production of formazan, but the absorbance is going to be less in comparison to the first condition, right? But if there is optimum superoxide dismutase present in the solution, then there will be negligible production of formazan, which indicates that the superoxide radicals are completely neutralized by these, the superoxide radicals present in the reaction mixture are completely neutralized by superoxide dismutase. Okay, so this was the overall overview or the principle behind the reaction method. Now we will discuss about the protocol. Now here I am expecting that if you are going to measure the superoxide dismutase activity in plant, then the first thing is you will have to collect the samples, right? You will have to collect the leaf samples, then you will have to freeze them immediately in liquid nitrogen and store them at minus 80 degrees Celsius until you are carrying out the reaction, right? If you are using the frozen samples, then you will have to thaw them on ice just before the extraction procedure. Okay, so for example, if we consider that you have collected the leaves, right? You have taken out the leaves. Okay, then firstly you will have to weigh them like 0.5 gram you can weigh, that is 500 mg, and you will have to homogenize them. Okay, you will have to simply crush them in potassium phosphate buffer. Okay, you will have to simply crush them in 100 millimolar potassium phosphate buffer of pH 7.0. Many a times when we are reading different papers, we see that they are crushing the plant leaves, or they are carrying out the every experimental uh, activity which is related with plants are generally carried out in buffer, which are also termed by extraction buffer. And if you see that their composition, it will consist of different salts like EDTA, PMSF. These kinds of salts are also present in these extraction buffer. But you will have to consider that whenever you are trying to calculate the enzyme activity from the leaves of the plant, you will have to remember that many a times these enzymes get inhibited by these kinds of salts. Okay, many a times they are not, but it is a general thing that if you are using different salts, like EDTA is generally considered as inhibitor of several enzymes. Okay, so that's why you can modify the procedure accordingly. Means initially you can carry out the process simply by crushing the leaves in 100 millimolar of potassium phosphate buffer, which is of pH 7. Okay, pH 7 is mandatory to keep the condition in neutral. Okay, now after crushing them, you will have to centrifuge it at 5000 rpm for 10 minutes at 4 degrees Celsius, and then simply collect the supernatant for measurement of enzyme activity. The overall procedure should be carried out in a manner that the temperature of 4 degrees Celsius is maintained. For example, if you are collecting the supernatant, try to collect it at 4 degrees Celsius, then try to maintain the experimental setup at 4 degrees Celsius. Okay. Now, so you have done with the sample preparation. Now, this sample which you have prepared, for example, the one will be controlled, in which there is leaf extract from normal plant, and then the another one will be treated, means in which you have treated the plants to stress condition or abiotic stress. Okay, similarly you can take your different samples, like if you are treating it with your own treatment like bioformulations, then you can consider those two. Moving on next, the second thing is the reaction mixture preparation. Okay, so for the determination of superoxide dismutase activity, you will have to take 3 ml of the reaction mixture. Okay, so the reaction mixture will comprise of phosphate buffer. Okay, firstly you will have to prepare 40 millimolar of phosphate buffer of pH 7.8. Okay, and in this phosphate buffer you are going to add 75 micromolar of nitro blue tetrazolium, then 13 millimolar of methionine, then 2 micromolar of riboflavin, okay, and finally the 100 micromolar of enzyme extract or the test solution in which you have whose enzyme activity you are targeting. Okay, so in this way you can see that this is going to be a reaction mixture. You will have to take 3 ml of this reaction mixture. Okay, now what you will have to do, you will have to simply place the mixture beneath the source of light. Okay, you can simply place it um, under fluorescent lamp, okay, for around 30 minutes. Okay, after 30 minutes you will have to measure the absorbance at 560 nm, because we have seen that upon exposure to light there will be production of formazan, which was blue colored complex. Okay, so it is going to show absorbance at 560 nm, okay, against a blank which is prepared by keeping the test tubes in dark condition. Okay, on the other hand, you are going to prepare your blank solution, which will be consisting of all the solution except your test solution. Okay, there is no need to add your test solution to this blank tube. Okay, only these substrates will be present. Okay, and keep it in dark condition so that there is no blue colored formation. Okay, now if you want to compare your results, means if you want to compare your test solution results with the standard, means if you want to calculate the concentration by comparing it with the means if you want to calculate the concentration, or if you want to calculate, for example, here you can see that formazan is produced and you want to calculate its concentration, right? So you can calculate it by comparing it with the standard. Okay, from the standard graph of formazan you can calculate its concentration which is produced in this reaction mixture, okay, of the test solution. Now for the preparation of standard solution, it can be simply prepared by dissolving the formazan with DMSO and 2 molar of potassium hydroxide in 1.6:1 ratio, okay, and diluting up to desirable concentration immediately prior to use. Okay, so you can also prepare a standard graph for comparison of your results. Okay, once you have taken the absorbance, you will have to simply calculate it. In here we are going to convert the absorbance change to a concentration change, okay, because the absorbance we are going to carry, means the absorbance we are going to calculate only for the determination of formazan production, right? So that's why we are going to convert this absorbance change to a concentration change by using the millimolar extension coefficient of superoxide dismutase. Okay, in this case superoxide activity if you are measuring, then you will have to use the unit per mg protein. Okay, means unit means the micromolar of substrate which are going to get converted to product per unit time. Okay, it is the enzyme activity unit per mg of protein, means we are going to compare it with the per mg of protein which is present in the reaction mixture. Okay, means per mg of protein means the per mg of protein per mg of enzyme. Simple, in simple sense, the per mg of enzyme which is present in the reaction mixture. So firstly, the absorbance of blank will be neglected from the absorbance that is given by the sample. Okay, then this is simply absorbance change. Okay, so first you will have to calculate the absorbance change, then you are going to multiply it with the volume of reaction mixture. For example, in this case we have taken 3 ml of the reaction mixture, then you are going to divide it with the time, means what is the incubation time in minutes. Okay, so we have taken 30 minutes. Okay, and then we are going to multiply it with the molar extension coefficient, that is 0.01. okay, so this is going to give you the results in unit per mg protein for the final superoxide activity determination. Okay, So also one thing is there that 1 unit of superoxide dismutase is reported to show 50% inhibition of formazan formation. Okay, so likewise you can calculate your activity. Okay, so uh, in this way you can carry out the experiment and you can calculate your result. Okay, now there is also one thing that when you are calculating enzyme activity, at that time you are concerned with this thing that how much substrate is getting converted into product per unit time. Okay, and here you can see that we are dividing the enzyme activity with the mg of protein, means we are simply calculating its specific activity. Okay, specific activity given by superoxide dismutase. Okay, I hope you understood the concept as well as you understood the protocol and calculation. Still, if you are having any kind of doubts, you can ask. And if you are going through your experiment, for example, also one thing I needed to mention, that if you are following a particular plant, okay, and you are in a process of carrying out the experiments related to determination of enzyme activity, then it will be better that first thing it will be better that you go with different research papers related to your targeted plant. Okay, first of all, it is necessary to know your plant very well. Okay, right, like what are the growth conditions and what are the optimal conditions and what kind of responses is given by those plant under different conditions, like, right. So for example, many a times what happens that not a single method cannot be employed for different plants. Okay, for example, if you are going through one plant, it may give different results, like if you are using buffer of different pH, then it can show different results, and if you are altering the pH, then it is going to show different result. Okay, the pH or the solution or the compounds which are present in the experiments matters. Okay, so you will have to keep it under consideration. Also, while collecting samples, you will have to consider different things, important things to maintain the temperature properly at 4 degrees Celsius is very much important while carrying out these kinds of experiments. Okay, further I'm going to try to make videos regarding other enzymes which are related for which are given by plants, like catalase or phenylalanine ammonia lyase, like these kinds of enzymes I'm going to consider for my further videos. Okay, so keep studying, and still if you are having any doubt, you can ask in the comment section. And also, if you are going through, if you have studied different method, if you have studied different method by from different research paper, you can also mention those their titles you can also provide in the comment section, so that other students may get benefit from that. Okay, thank you so much.